Imagine a single unifying technology that can perform thousands of medical diagnostic tests simultaneously in the palm of your hand. Imagine a disposable test strip, able to detect cancers, viruses, bacteria, antibodies, and many other biological targets. A device that synchronizes with a phone or tablet, allowing for immediate diagnosis. A device that consumers can connect to their phones and use every day. At home to test their kids for the flu, or at the doctor's office to get a complete diagnosis. At the grocery store to check fruits and vegetables for E. coli and other pathogens. In the garden to check the health of their plants. A device that synchronizes with the cloud, where data analytics can track the progression of diseases, the health of animals and plants in the food chain, and environmental conditions globally. All this for the cost of a cup of coffee. Fluids can come from any natural source. Blood, saliva, animals, plants, and the environment. Let's say we want to detect the presence of HIV. A simple blood drop flows through a disposable test strip, landing in a reservoir containing TPG's tiny solid-state nanopore sensor, comprised of 2 concentric nanopores. When voltage is applied, all molecules are pulled through and current impedance signatures indicate their presence. To detect any target of interest, such as this HIV protein, TPG has created a synthetic polymer scaffold designed to capture only that target. As the fluids interact with the scaffolds, the target analytes are captured. As molecules are pulled through, we can tell which are our scaffolds. Here is the label. The signal shows the HIV protein is bound. If there was no protein, the bump would be missing. The targets are counted, while all the background molecules are ignored. Any molecule along the molecular spectrum can be captured. One only needs to change the molecule that captures the target of interest. Here, we detect and count influenza. The events are then analyzed to determine concentration levels. The data is then encrypted and sent to a smart device, where third-party apps make a clinical diagnosis. Each test can be placed on its own test strip. We can also detect the presence of DNA sequences, indicating the presence of bacteria, viruses, cancers, and even particular genes and gene mutations. To do so, we create synthetic probe molecules that bind directly to those target DNA sequences. As before, we introduce serum into the test strip. If a target mutation exists in someone's DNA, our probe will bind to it. Here we see the probe has found the gene mutation of interest, yielding a positive test result. To detect any gene, we only need to create a probe for that unique DNA sequence. These tests can also be placed on test strips separately. Multiple tests can be performed at the same time by using multi-strip hubs, and hubs can be daisy-chained for further extensibility. Test strips can also have multiple chips, improving time to results and test sensitivity. And all of it can be run using off-the-shelf microprocessors, allowing for an infinite range of inexpensive consumer or professional electronics devices that can be mass-produced for everyone everywhere. This method is faster and more accurate than conventional diagnostic devices, and because no other chemistries are necessary, TPG products far outperform ELISA and PCR tests in both accuracy and cost of operation. This modular system can be sold in retail stores for simple and inexpensive access. Consumers can now test for most health conditions on a daily basis, and doctors can perform tests for genetic disorders, cancers, or more complex diagnosis. TPG devices are exact and work precisely the same way for any and all tests. The end user sees the correct results every time. The cost of TPG devices and strips are so low, it would relieve the financial burden of healthcare from the government and private carriers, not to mention helping the uninsured. The more we know about ourselves and our world, the better the opportunity to improve it. Two Poor Guys makes all this happen.